मेरा 12 बजे तक है ये क्लास, लेकिन उसके बाद सर बोल रहे कि डेमो भी है। ठीक। Sejal, am I audible, Sejal, clearly? Hmm? Okay. So last lecture we have studied till hmm? Okay. Wait, let me share the screen now. It will take 1 to 2 minutes because again it is PowerPoint presentation. Wait. Sejal, I'm going to share the screen now. You have to tell me whether it is visible or not. Okay? Hmm? Yes. Is it visible, Sejal? Yes. Okay. Okay. So I think a pollination we have done, right? Hmm? Tell me yes or no. Till pollination we have discussed now, Sejal. Hmm. Yes. Okay. Hmm? Okay. Now we have to discuss about outbreeding devices. Hmm? Now, in this, we are going to talk about double fertilization also. Hmm? It is there in your textbook. Hmm? So, data. Okay, so outbreeding devices. I hope you are aware of this term breeding, right? Many plants have a mechanisms that discourage or prevent self-pollination. Okay, see, last lecture we have talked about adaptations in each and every if it is pollination by bats, if it is pollination by birds. So in each and everything we have studied about modification, isn't it? So many plants have mechanism that discourage or prevent self-pollination. So to promote cross-pollination and increase the genetic diversity. What do you mean by genetic diversity? Look here. Diversity, you know what is diversity? Variety among the living organism. We are talking about genetic diversity here. So to promote cross-pollination and increase the genetic diversity. Plants have evolved a wide variety of sexual strategies. Like genetic diversity is essential for evolution, of course. I told you before also, if it is a sexual mode of reproduction, exchange of gametes, exchange of genetic material is there, and with the help of this, you will get variations, right? So जितना diversity ज़्यादा होता है उतना वो it is important for the process of evolution this to know. So it is by natural selection. It is by natural selection only. You know the meaning of natural selection. What is it? Natural selection. Those who are fit to the environment. Nature is going to select only those organisms, isn't it? So, uh, continuous self-pollination can cause inbreeding depression. Now, you will ask me, ma'am, what is inbreeding depression? See, what happens here? Self-pollination, that means we are going to consider only a single plant in a or a single flower. So, because of this, what will happen? Yes, genetic stability is maintained. Yes, you are correct. But after some generation, what will happen? Tell me, what will happen after some generation? फर्टिलिटी जो होती है वो बहुत कम हो जाती है एंड दिस इज कॉल्ड एज इनब्रीडिंग डिप्रेशन गॉट इट so genetic diversity is essential. Plants have developed many devices to encourage cross pollination. And here some examples they have given. So examples of outbreeding devices are unisexuality, dichogamy, then you have prepotency, then you have heterostyly, hercogamy, and self compatibility, right? So these are nothing but some of the example where you will come to know how plants have developed many devices to encourage the cross pollination. हाँ, प्लांट ने खुद को कैसे डेवलप किया है to avoid self-pollination and to promote cross-pollination. Okay, Sejal, understood? Tell me, what is the need or importance of outbreeding devices? Here we are going to promote cross-pollination and to avoid self-pollination. Why? Because of self-pollination, inbreeding depression is there. Fertility after after 2 to 3 generation, fertility जो है वो बहुत कम हो जाती है, ठीक है? So in this, to avoid the self-pollination and to promote the cross-pollination, there are some outbreeding devices that we have to study now. So unisexuality, one by one we are going to talk now. Yes. Now what is unisexuality? In this case, the plant bears either male or female flower. And it is called as dioecium. Flower are unisexual. You know the meaning of unisexual, bisexual. Self-pollination is not possible in this case. What we are doing, we have to avoid self-pollination. We have to promote cross-pollination. Plants may be monoecious or it is dioecious. For example, maize, papaya. Here you can see. Now, understood about this? First one. Then you have dichogamy. Dichogamy, the stamens and carpels of bisexual flower mature at different times. Stamens भी है, that is your male androecium, carpels or female female androecium. Sorry, female gynoecium, female, or we can say gynoecium. So stamen male, carpel female. Of a bisexual flower mature at different times, and hence cross pollination becomes inevitable. Okay. So please keep this in your mind. Plants either protandrous like sunflower or protogenous like example they have been given gloriosa and mycelia. Now, what do you mean by protandrous or protandry and protogeny? Tell me, Sejal, do you have any idea? See, protandry means what? In this type, androecium matures earlier than the gynoecium. Androecium जो है ये पहले, because here in first sentence only you will come to know it is bisexual flower. Correct. That means both stamens and carpel. Present in a single flower. But if you talk about maturity, both stamens and carpels mature at different times. तो एक तो पहले होगा और एक बाद में होगा. Like this only the situation will be understood. So if it is protandry means what? Male organs gain reproductive activity before female organ. Huh? That means in other sentence what we can say? Androecium matures earlier than the gynoecium. इसमें क्या होता है? एंड्रोशियम मैच्योर होता है पहले. Now second, protogeny, exactly opposite. In this type, gynoecium matures earlier than the androecium, and that's the meaning. Stamens and carpels mature at different times. Okay, understood. For one mark in your exam, they may ask you what is protandry and protogeny. So from this sentence, pro means before, proto, gyne or gyne, or protandry, that means before. क्या होता है? Mature before female organ, male organ gain reproductive activity. Like this, you can remember. I hope it is clear from your side now. We are talking about how to promote cross-pollination to avoid self-pollination, because there are some disadvantages of having the self-pollination. After some generations, after 2-3 generations, fertility is lost. Got it? So to avoid the self-pollination and to promote the cross-pollination, there are certain devices, many devices to encourage the cross pollination, and we are talking about those devices only. Okay. Sejal, I need your reply. Understood? You can, yes, you can write. Okay. Thank you so much for your reply. Then second device, third one is prepotency. Prepotency, pre again, before pollen grains of other plants germinate rapidly over the stigma than the pollen grains from the same flower. For example, apple. Very simple, no need to explain also. Pollen grains of other flowers germinate rapidly over the stigma than the pollen grains from the same flower. Because what we are doing, we have to avoid self-pollination. So we have to ignore same flower. Okay, and we have to concentrate on other flowers to encourage the cross-pollination. Then you have heterostyle. Now, heterostyle, and it is also called as heteromorphy. It is given in your textbook. Now, this facilitate cross-pollination. A pin-eyed or long-styled flower and short stamens. Short style flowers have short style and long stamens. See what kind of modifications they have given to promote cross-pollination. It is really very interesting. Pollination occur between flower of same length of style and the stamens. An example is also given. Understood? Heterostyle, and it is also called as heteromorphy. So this prevents the pollen from reaching the stigma and pollinating it. In heteromorphic plants, pollen grains produced from anther, pollen grains produced from anther pollinate stigmas produced at the same level. So in this way you can remember. Then you have herkagamy. Hmm? Herkagamy. It is a mechanical device again to prevent self-pollination in a bisexual plant. Yes. In plants, natural physical barriers are there. It's present between two sex organs and avoid contact of pollen with stigma of same flower. That means what happens? We have some physical barriers. Physical barriers to prevent करते हैं. In plants, there are some natural physical barriers present between two sex organs. Two sex organ means what? Sejal, here we are going to consider stamen and the carpel. That means pollen grains and the stigma, and avoid contact of pollen with stigma of the same flower. तो इसमें क्या होता है, अगर सेम फ्लावर में ही if the plant is dioecious, if stamen and what we can say carpel present in the single flower, here we need to avoid self pollination. So in some plants what happens, there are some natural barriers जो prevent करते हैं self pollination. Why? Understood? Huh? In some plants, natural physical barriers are there. Calotropis, for example. And the last we have self incompatibility, that is self sterility. Because sterility जो है, you know about it, right? Sterile, sterility. You must have heard about these terms. Okay. So in self-pollination, same thing is going to happen. After a few generation, 2 to 3 generation, जो है वो sterile हो जाता है plant. उसकी fertility बिल्कुल भी पूरी निकल जाती है. So in this case, it is not beneficial. So that we have to avoid the self-pollination. This is one of the disadvantages of having self-pollination after 2 to 3 generation, and this is called as inbreeding depression. I have already explained you, and that's the reason we are talking about some devices to encourage the cross pollination, right? So now this is a genetic mechanism due to which the generation of pollen and the stigma of the same flower is inhibited. Got it? For example, tobacco and the sera. Okay, understood? I hope it is clear now.
Yes, sir.
Very simple. So we have talked about outbreeding devices to avoid self-pollination. What is the cause of self-pollination? It is inbreeding depression. And to promote, or we can say to encourage cross-pollination, we have talked about several devices, how it works exactly to encourage cross-pollination. Understood? Now next is pollen pistil interaction, and please give triple star to this because this is very frequently asked question. You have to keep this in your mind. Now what is pollen pistil interaction? From this, what you have understood? Tell me, Sejal, what is given? What do you mean by pollen pistil interaction? Exactly what happens in this? Give me your answer in one sentence. Yes. What is it?
Pollen grains interact with stigma.
Pollen grain interacts with stigma. Okay. Anything else? Interaction. Okay. Correct. Anything else? Okay. You are correct. It is the interaction of pollen grains with stigma. Right. It begins with the pollination, of course, and ends with the fertilization. Why? Because after fertilization, development begins. This you know, we have different developmental stages. Consider our example only, human being example. Our fertilization is internal. Plus, we have different developmental stages. Fertilization. In fertilization, there is fusion of male and female gametes. Am I correct or wrong? Tell me. आफ्टर फर्टिलाइजेशन सेगमेंटेशन इज देयर, जो जाइगोट बनता है उसका सेगमेंटेशन होता है. uh, 1 to 2 cell, 2 to 4 cell, 4 to 8, 8 to 16, 16 to 32. Then morula formation, then what we have, blastula, then gastrula, where three germ layers are going to produce. Then implantation. You must have heard about different developmental stages. And in case of this also, zygote, then zygote converts into embryo, then fetus, and then the infant. Right? So try to remember all these things, right? Now it begins with pollination and ends with fertilization. You know the meaning of pollination. Pollination means transfer of pollen grains from anther to the stigma, and fertilization where it is going to fuse. So fusion is nothing but fertilization only. Understood? So it is the interaction of pollen grains with stigma. It begins with pollination and ends with fertilization. All the events from deposition of pollen grain on stigma, to entry the entry of pollen tube in the ovule are called as pollen pistil interaction. Understood till here? Pollination does not guarantee the transfer of right type of pollen land on stigma. The pistil has ability to recognize right pollen. Underline this and keep this in your mind. हाँ, and accept the pollen of the same species. तो उसको एक ability होती है. Pistil means what, Sejal? Carpel. That means it is female reproductive part. Where you will see stigma, style and ovary. Ovary में जब ये introduce होता है, जब ये enter करता है, then there is a cell. हाँ, जहाँ पे fertilization होता है. So pistil has ability to recognize. So wrong type of pollen discarded by pistil. Okay. So pistil has ability to recognize right pollen, and accept the pollen of the same species. So the wrong type of pollen discarded by pistil automatically. So compatibility and incompatibility of pollen pistil is determined by the proteins. कि क्या अच्छा है क्या नहीं है. This process involves pollen recognition followed by promotion of, और promotion होगा या inhibit होगा. That means activate होगा fertilization process या stop होगा. It is decided by pistil only. हाँ, or inhibition of pollen. Okay, so a physiological mechanism operates to ensure only intra-specific pollen. Now, what do you mean by intra? Intra means within the same species. Huh? Pollen germinates successfully. If it is within the same species, then it is called as intra-specific pollen germination. What we can say? The process is successful then after. Why? It's only because of recognition. ये जो after a recognition, what will happen? It is going to decide whether to promote the process of fertilization or to inhibit the process of fertilization. Understood? So the compatible pollen absorb water, nutrients from surface of stigma, germinates and produce pollen tube. And then there is formation of pollen tube. ये पोलन ट्यूब फॉर्म होना पड़ता है ताकि वहां से पोलन ग्रेन ट्रांसफर हो जाए और यहाँ से वो इंट्रोड्यूस हो जाए एक सेल में, which is there in the ovary. Ovary is swollen part of the pistil. This you know, stigma, style, and ovary. So its growth through the style is determined by again specific chemicals. The stigmatic surface provides essential prerequisites for successful germination. See how it is. Huh? For each and every line, for each and every step, you will come to know how exactly it works. हाँ, सबसे पहले तो recognition होता है. हाँ, then only pollens are accepted by stigma. हाँ, so pistil has that kind of ability to recognize right pollen. Understood? If it is wrong, it is discarded. Understood? Then there is physiological mechanism. Then compatible pollen absorb water and nutrients. Its growth through the style is determined by specific chemicals again. So the stigmatic surface provides essential prerequisites for successful germination which are absent in pollen. So pollen tube finally pushed to the ovule and reach the embryo sac. Just now I told you. Then it enters into the ovary for further process or for the fertilization. So the tip of the pollen tube enters the one synergate. Now try to remember last say diagram which we have discussed in last session, anatropous ovule. Please try to remember. In that we have studied development of female gametophyte. How many have four haploid megaspores are going to release? Out of four, three are generated and only one megaspore or embryo is there. Then growth is nourished by nucleus. Try to understand. Then with the help of mitosis there is post division, उसका दो बनता है फिर चार, and this is how. So after that third mitosis division is also there, in which eight nucleus are going to form. Two polar nucleus, three antipodal cells and one female gamete. And besides that one female gamete, you have two synergids. Understood? Try to remember the diagram which we have discussed before. Sejal, yes or no? Huh?
Yes, ma'am.
The tip of the pollen tube enters in one synergid and rupture to release the contents. Yes, this diagram. So here you can observe pollen grain. This is your pollen grain. Huh? Stigma. Huh? This part is stigma. Male germ cell from here. Huh? This is pollen tube formation. Pollen tube से transfer होगा ये. Then ovary. This is female germ cell which is inside the ovary. From here it enters. So this is the germination of pollen grain on stigma. Very simple diagram. So this is pollen pistil interaction. Understood? Hmm? Pollen pistil interaction. So go through the diagram carefully. From this diagram, you will come to know how it is going to form and how it enters into the ovary for further fertilization. Right? Okay. Now, artificial pollination and the hybridization. Artificially, how it takes place. Now, artificial pollination. In this, you can see it is with the help of mankind activities. Hybridization means what? If you select two characteristics of two different plants, and whatever you are going to get in next generation will be carrying characteristics of both the plants. That means hybrid variety, we can say. So this can be activated through man-made activities or with the or we can say artificial pollination. So help in the plant improvement program, artificial pollination. A breeder is often interested in crossing different species. Just now I told you, if you select two different plants with two different categories in next generation you will get characteristics of both the plant. So you have to combine the desirable characteristics, or we can desirable characteristics means what? The characteristics or the characteristics in which you are interested. Producing superior varieties. So that superior variety is actually a hybrid variety. So breeder wants to cross a particular plant with a desired pollen grain. So artificially this can be done. So a bisexual flower is artificially transformed into the female flower. And this is done by emasculation. Underline this or write down in your notebook. Emasculation is the removal of stamens or the anthers. This is artificial method only. Now in this diagram you can see emasculated flower is immediately bagged. Can you see in this diagram which is also called as bagging procedure? Here can you see emasculated flower जहाँ से pollen grains हमने निकाले हैं, remove किए हैं. जब हम वो करते हैं, वो प्रोसेस हम करते हैं, immediately it is banned to avoid pollination, further pollination by unwanted pollen grains. ये होना नहीं चाहिए आगे, अनवांटेड पोलन ग्रेन्स आ सकते हैं, चांसेस है, बिकॉज वी हैव डिफरेंट एजेंट्स. Understood? So this is called as bagging process. In this diagram, you can observe, and here you can observe how it is like how are you going to remove the stamens or the anther. And in this diagram, followed by this, immediately bagging process is done to avoid unwanted pollen grains, to avoid pollination by unwanted pollen grains. Clear?
Yes, ma'am.
Artificial pollination and the hybridization. Yes. Now, double fertilization. Please give triple star to this. Double fertilization. This is very important. It, again, very frequently asked question. And what you can do is, along with me, like while explaining only, you can take notes in your own sentences. But please, you have to draw diagram also, Sejal. Okay. I will tell you how to draw diagram. It is very simple. You can take help of anatropous or ovule, which we have already studied. So you can go through the diagram once and try to draw diagram. A double fertilization. Now, why the name is given double fertilization? Do you have any idea? Why the name is given double fertilization?
Because fertilization is taking place two times.
Correct. See, in our like in case of human beings, like take a human being example only. It is internal fertilization. This you know. There is fusion of male and female gamete. One male gamete and one female gamete. When they fuse together, there is formation of zygote, right? But in double fertilization, you have two male gametes. Understood what I'm saying? So one male gamete fuses with one female gamete to form zygote. Huh? Now this zygote. Now here procedure. If you talk about it, double fertilization, then there is a fusion of second male gamete with the, what is it? Could you please tell me? Syngamy we called it as, which is a triple fusion basically. Any idea? The one process which is very simple, haploid male gamete with haploid female gamete to produce a diploid zygote. Haploid male gamete with haploid male gamete and the female gamete. When fuses with each other, there is formation of diploid zygote. ये हो गया एक fusion. But we have two male gametes, right? But what happens in triple fusion? Second haploid male gamete fuses with diploid secondary nucleus. Okay. So you can write it down, Sejal, huh? Then only you will come to know. Later on, you can. I'm going to give you 5 minutes to draw diagram and to write in front of diagram only or below the diagram only. Write down this. In this way only you have to write. Sejal, look here. Uh, One male gamete, which is haploid in nature. So in bracket, you can write down single N plus female gamete, that is egg. Again, you can write down single N. Gives diploid zygote. So write down, give one arrowhead, write down zygote, and in bracket 2N you can write down. Then second line, what you can write is second haploid male gamete. We have two male gametes, right? That's the reason double fertilization we are talking about. Huh? Secondly, in second line, you are going to write about second haploid male gamete fuses with diploid secondary nucleus, and one more arrowhead you have to give. This will form primary endosperm nucleus. Now, why endosperm? ये endosperm form कब होता है? हाँ, क्यों होता है? एंडोस्पर्म का रिजल्ट कब आता है? देखो, जब पहली बार first time when there is fusion of male gamete and female gamete to form zygote which is diploid in nature. इसको nourishment देने के लिए there is formation of endosperm for further development, right? If first fusion fails, then there is no formation of any endosperm. That means triple fusion is not going to take place only. Understood what I'm saying? What happens in first stage? See, there is fusion of male gamete and female gamete, N plus M to form zygote which is diploid in nature. After that, second male gamete when fuses with endosperm nucleus, primary and secondary nucleus, it will produce primary endosperm nucleus, that is PEN, pain. So this will provide, endosperm is nothing but nutrition only. So this will provide nutrition to the zygote for further development. Understood? So zygote, and that's the reason afterwards zygote develops into an embryo. It's only because of second fusion. And that second fusion is nothing but second haploid male gamete when fuses with diploid secondary nucleus, then there is formation of primary endosperm nucleus. Okay, and this endosperm is going to provide nutrition to the zygote for the further development. That means later on it develops into an embryo. समझ में आया सेजल मैंने क्या बोला?
Yes, ma'am.
And that's the reason we called it as double fertilization. If first step fails, like for example, if there is no fusion of male and female gamete, zygote is not going to form only. हाँ? जब प्राइमरी क्या होता है, प्राइमरी फ्यूजन कौन सा है, जहाँ पे मेल एंड फीमेल गैमेट फ्यूज होते हैं और जिसका रिजल्ट जाइगोट होता है. अगर ये जाइगोट ही नहीं फॉर्म हुआ, पहला ही स्टेप अगर फेल होता है, तो सेकेंड फ्यूजन भी नहीं होता है. वाई? क्योंकि जाइगोट होगा तभी तो एंडोस्पर्म फॉर्म होगा उसको न्यूट्रिशन देने के लिए. अंडरस्टूड? सो दैट इज देयर. नाउ लेट्स टॉक अबाउट इट. It is a complex process in angiosperm. It is also called as triple fusion. The process involves the joining of female gametophyte with the two male gametophytes. It begins with a pollen grains, others to the stigma of the carpel. The pollen grain germinates to form pollen tube that extend. This we have already studied down down to what the ovary through style. The tip of the pollen tube then enters the ovary, penetrates through the micropyle opening in the ovule. Now you know what is chalaza, what is micropyle. We have studied in anatropous ovule. Try to remember. Now, pollen tube may enter the ovule either from micropyle. If it is entering through micropyle, then it is called as porogamy, or from chalaza, then we call it as chalazogamy, or if it is through any part or integument, then it is called as mesogamy. So in this three diagrams, you will observe. If it is through micropyle, this is your micropyle, right? If pollen tubes enters through micropyle, then it is called as porogamy. If it is through chalaza, this is your chalaza. We are talking about anatropous ovule. If the pollen grain enters through chalaza, then it is called as chalazogamy. And if it is through any part or any integument, then it is called as mesogamy. Understood? Yes, in this they have given. You can write it down. Let me explain. First, I have already explained you, but still, the pollen tube reached embryo sac and absorbed watery substance from synergate or jade cell and release its contents. The pollen tube contains two male gametes. This you know. पोलन ट्यूब में दो मेल गैमेट होते हैं. One haploid male gamete fuse or combine with haploid egg cell to form a zygote. Here you can see male gamete which is haploid in nature, egg nucleus, or egg gamete or female gamete. Female gamete, here you female gamete, zygote will form, and zygote then after converts into embryo. But when? Sejal, when? Huh? This is my question. When we have different developmental stages, after zygote, the next stage is embryo. Hmm? This process is called as syngamy. Very simple. Then second male gamete, we have two male gametes. Here it is written two male gametes. In pollen tube we have two male gametes. एक मेल गैमेट एक फीमेल गैमेट के साथ कंबाइन होकर जाइगोट फॉर्म करता है. This process is called as syngamy. And second male gamete fuses with polar nuclei, which is 2n. So it gives PEN, which is triploid. That is primary endosperm nucleus. Okay, and this is called as triple fusion. Now this endosperm, primary endosperm nucleus is going to provide nourishment to the zygote. This will give nourishment to the zygote, and then after this zygote converts into embryo. Understood? Huh? So syngamy plus triple fusion is called as double fertilization, which is a unique feature of angiosperms. हाँ, तो मैं क्या एक्सप्लेन कर रही थी आपको, ये मेल गैमेट फीमेल गैमेट के साथ जब कंबाइन करता है, लेकिन अगर जाइगोट नहीं फॉर्म होता है, अनफॉर्चुनेटली इफ fertilization fails here, then what will happen? Is there any formation of primary endosperm nucleus? Of course not. ठीक है, because इसका मतलब क्या है, ये प्रोड्यूस क्यों हो रहा है? We have one more male gamete. That's the reason. Yes or no, tell me. And secondly, this primary endosperm nucleus is going to provide nourishment to the developing zygote, which converts into embryo. Understood? So this is how it works basically. Okay. Understood, Sejal? So this simple process is called as syngamy. Then when male gamete fuses with polar nuclei where endosperm is going to form, this is called as triple fusion, and syngamy and triple fusion collectively called as double fertilization, which is a unique feature of angiosperms. So quickly write it down here given. The one haploid male gamete fuse or combine with haploid egg cell to form zygote called as syngamy. The second male gamete fuse or combine with two haploid polar nuclei of the large central cell called triple fusion, and the primary endosperm nucleus which develops into triploid endosperm. Here in this descriptive part, they have not given everything which I have discussed just now with you. ये एंडोस्पर्म जाइगोट को नरिश करता है, उसको न्यूट्रिटिव वैल्यू देता है ताकि जाइगोट आगे जाके एम्ब्रियो में कन्वर्ट हो जाए. लेकिन अगर ये प्रोसेस ही नहीं होती है, जाइगोट फॉर्म नहीं होता है, तो इसका कुछ मतलब ही नहीं है. ठीक है, तो दिस प्रोसेस ऑल्सो फेल्स, or this process is not going to happen if this post fertilization fails. Then embryo is not going to form. So this is how it works. So quickly write down in your notebook, because this whole thing will give you an idea how double fertilization is there, actually exactly how it works basically. So I hope it is clear from your side now. Please give me your confirmation. Yes, ma'am. No, ma'am. So both the male gametes participate, and therefore it is described as double fertilization. Now, what are the, yes, first you write it down, Sejal, and then we'll be talking about significance of double fertilization. Please. You have to draw diagram also. Draw only one diagram, porogamy का, जहाँ से micropyle से pollen grain enter करता है, or sorry, pollen tube enter करता है. So draw only one diagram, because in your textbook 3 diagrams are given. One is porogamy, chalazogamy, and the second or third one is mesogamy. So you can draw only single diagram, porogamy. Please draw and write down this one. Yes or no?
Yes, ma'am.
Please do it. I'm going to give you 5 minutes. Please do it. on priority only because this is very important. Hmm?
Done, ma'am.
Any doubt in double fertilization, Sejal? No, na?
No, ma'am.
Okay. Hmm. Yes. Next is your mouse is not working on screen. Yes. Life cycle of flowering plants. Flowering plants. Hmm? So let me see if it is given in your syllabus diagrammatic representation of double fertilization. Hmm? You just go through this, Sejal, because we have already discussed. Triploid primary endosperm nucleus repeatedly divide mitotically to form a nutritive tissue. We called it as endosperm. In post-fertilization changes within the ovule, embryo and endosperm are seen to develop simultaneously. So other cells of embryo sac um, is also given. Formation of triploid endosperm, trigger cell division which leads to formation of endosperm. So there are three types of endosperm on the basis of mode of development, how it develops. Hmm? Nuclear type, cellular type, and halobial type. Hmm? Go through the diagram. It is very simple. We have already studied it. From here you can start. हाँ, यहाँ से आप start करते हो कर सकते हो pollination anther. You take a section of anther. This is how it looks. We have four pollen sacs in which pollen grains are present. In each pollen it is bilobe structure. In each lobe two pollen sacs are there. Then See here, you can see pollen grain, which is haploid in nature. Microgametophyte, pollen grain, stigma, sperm cell, tube cell, nucleus. Then formation of pollen tube, egg cell. Here there is fusing, double fertilization, we can say. Then zygote then enters into embryo. Then seed coat, endosperm is going to form, which is triploid. So this is angiosperm life cycle. Done? Okay. Now we have to study the significance of double fertilization. Significance. It is a unique feature of angiosperm. It ensures that the parent plant is a seed with a food store only if the egg is fertilized. Hmm. Now the diploid zygote develops into an embryo and then it develops into a new plant. The triploid PEN. What is the full form of PEN? Sejal, you must have written just now. What is the full form of PEN?
Primary endosperm nucleus.
Yes, very good. Primary endosperm nucleus develops into nutritive endosperm tissue. I have already discussed this. हाँ, एडवांस में ही मैंने आपको ये डिस्कस किया था. एंडोस्पर्म मीन्स व्हाट? न्यूट्रिटिव वैल्यू ओनली. So nutritive endosperm tissue, it restores the diploid condition of plant by the syngamy process. It also helps to avoid polyembryony. What are post fertilization events? ये तो हो गया fertilization. We have talked about pre-fertilization events. Then fertilization events, that means double fertilization and everything we have studied. Now we have to discuss about post-fertilization events. That means what is the future? जैसे you know that a flower develops into fruit and ovules into seeds. So we are going to talk about that now. What are the major events of post-fertilization events? Development of endosperm, development of embryo from diploid zygote. Yes, zygote then after develops into embryo. This you know. कब होगा develop? जब उसको endosperm nutritive value provide करेगा. That time zygote will convert into embryo. So these are post-fertilization events. Development of endosperm, development of embryo from the diploid zygote. Development of seed from ovule and development of fruit from ovary. So flower में you know that ovary, sorry मैंने flower बोला था, flower मतलब मुझे ovary बोलना था basically. Ovary develops into fruit and ovules into seed. So these are post fertilization events. Understood? Now next, yes, development of endosperm. First post fertilization event we are going to talk. The pain starts dividing rapidly. It forms a food laden tissue which is used during development of growing embryo in the seed. That means same sentence, same thing I'm going to repeat again and again. Endosperm gives nutrition to zygote, which then after converts into embryo. Same thing they have given here also. In angiosperm, endosperm is a post-fertilization product, that is 3N. In gymnosperm, it is pre-fertilization fertilization formed and it is haploid in nature. So there are three types of endosperm on the basis of mode of development. कैसे वो develop होता है? On the basis of mode of development, there are three types: nuclear, cellular, and cellular. So we will see what is it. Yes, nuclear endosperm. It is observed in 161 angiosperm families. In the nuclear type of endosperm development, the primary endosperm nucleus divides by repeated mitotic pre-nuclear division without the formation of wall and produce large number of nuclei. A big central vacuole is also there. Push all nuclei towards the periphery. Then wall forms between nuclei. For example, wheat, sunflower, and coconut. So this is nuclear endosperm. कैसे develop होता है? Based on that, on the basis of mode of development. First is nuclear endosperm we have studied. Second is cellular type. Now in cellular type endosperm development, the primary endosperm nucleus divides mitotically and it is immediately followed by wall formation. So that endosperm is cellular right from the beginning. It is mostly observed in 71 families of dicot plant. Of dicot plant. Example also given. Now, the next one, it occurs in the order Helobioles of monocot. It is intermediate of nuclear and cellular type. Yes, दोनों के बीच में ये हम consider कर सकते हैं. It is intermediate of nuclear and cellular type. After first division of primary endosperm nucleus, it is followed by transverse wall which divides the cell unequally. The smaller cell called chalazial cell and the larger cell is called as micropylar cell. Example is also given asphodilus. थोड़ा सा याद करना पड़ेगा because the name itself give you the meaning. Nuclear endosperm, cellular endosperm. Like this you can remember. Now second post yes, second one is development of embryo. The process of development of zygote into embryo is called as embryogenesis. Genes, embryo. You have to split the word. Embryogenesis. Genesis is nothing but formation. Formation of embryo. So the process of development of zygote into an embryo is called as embryogenesis. The embryo develops at the micropylar end of the embryo sac. I told you there are three ways through which pollen tube enters. The one is orogamy, that means through micropyle. Second, you have chalazogamy through chalaza and mesogamy through integument. Understood? So embryo develops at the micropyle end of the embryo sac. The growth of embryo triggers means activate करता है उसको. The growth of embryo triggers only after certain amount of endosperm is formed. Of course, embryo की growth कब होगी? वो develop next stage में कब होगा? जब उसको nutrition मिलेगा. So endosperm is going to give this nutrition. Understood? So after fertilization, the embryonic development begins. I told you before also. Immediately after fertilization, development begins. Not only in a plant case, in case of human beings also, immediately after fertilization, development begins. Okay, the zygote divides to from proembryo, then larval cell towards micropyle is called as basal or suspensor initial cell, and towards chalaza end is called terminal or embryonal cell. So we have two types of cell, embryonal cell or initial cell. If it is towards the micropyle, if it is towards the chalaza, you have to remember in this way. Now suspensor cell divides, that means basal one, transversely and form 6 to 10 filamentous cells. The first cell of supersensor suspensor towards the micropyle and becomes swollen and function as underline this hostorium. Now lower is hypophysis. The lowermost cell suspensor, and suspensor helps in pushing the embryo in the endosperm, and the embryonal initial undergoes 3 successive mitotic division to form octant. Octant, sorry, octant. That means 3 successive mitotic division means what, Sejal? 1 to 2, ये first division हो गया. 2 to 4, second division. And 4 to 8, that's the reason name is given octant. So three successive mitotic divisions, 1 to 2, 2 to 4, 4 to 8. That's the reason given octant. Understood? The lower, here again some extra points they have given. Please go through this. Like protective sheath of plumule. You, ये तो आपको पता है ना, radical develops into root and plumule develops into shoot. Future organs I'm telling you. So protective sheath of plumule is called as coleoptile. Protective sheath of radical is called as coleorhiza. Finally, ovule is transformed into seed and ovary into the fruit. So these are different developmental stages, right? How ovary develops into fruit and ovules into seeds. So just go through this. It is very simple. Exactly, they have mentioned about overall development, what develops into what. Like this, you can remember. Hmm? Done?
Yes, ma'am.
Different developmental stages are there, Sejal. Okay, no need to worry about this because it is very simple. What develops into what? You have to remember. Like if ovule is transformed into seed, ovary into fruit. तो ये exactly कैसे होता है? We need to consider all the parts. कैसे जैसे we are triploblastic animals. गैस्ट्रोला में जब त्रिजम लेयर फॉर्मेशन होता है, जैसे हमारी किडनीज है, स्किन है, तो हम क्या बोलते हैं, स्किन इज एक्टोडर्मल इन ओरिजिन, मतलब गैस्ट्रोला में जो एक्टोडर्म हमारा फॉर्म होता है. So we have we are triploblastic animal. We are derived from three germ layers, right? During embryonic condition, there are formation of three germ layers which will decide the future organs. Huh? In this way also, if you study about development of embryo, you will come to know कि कौन सा part किसमें develop होता है. Like this, जैसे plumule मैंने बोला, तो प्रोटेक्टिव शीथ का प्लूम्यूल का प्रोटेक्टिव शीथ को बोला जाता है कोलियोप्साइड. So in this way, plumule develops into shoot and radical develops into root. So overall उन्होंने बोला है. Okay. Now next is your development of embryo. Okay. In this way, you can remember diagrams we have given here for your understanding. Stages in development of monocot embryo. Hmm, See, this is your egg, fertilized egg. Hmm, एक का दो में कन्वर्जन होता है. इट डिवाइड सेगमेंटेशन होता है, दैट मीन सेगमेंटेशन. 1 into 2, 2 into 8, 1 2 3 4 5 6 7 8, not 1 2 3 4 5 6 7 8, like this you can see. ये एक होगा शायद segmentation, because in human beings also segmentation is there. We called it as cleavage. Zygote is one celled, one का two celled होता है, 2 to 4, 4 to 8, 8 to 16, and then finally 32. Then there is arrangement of micro and mega cells. And then there is formation of gastrula, blastula, like that. So here also, if you see, you will observe. is your mature embryo with two cotyledons, one and two. Okay. In detail, they have given everything. Now, development of seed and fruit. You know that ovary develops into fruit and ovules present in the ovary develops into seed. Now, the goal of reproduction is to create offspring for the next generation. One of the ways that plants can produce offspring is by forming seeds. The flower must be pollinated in order to produce seed and fruit. Seed development is initiated by fertilization. We have already talked about this region. The integuments of the fertile ovule persist and get transformed into seed coat of mature seeds. Seeds sometimes consist of two distinct coverings, typical outer seed coat. Here you can observe in this diagram. Testa and the inner thin membranous tegmen. The nucleus of the ovule may be persist in some genera like black pepper and beet as thin papery layer. We called it as perisperm. So different coverings you can observe here. This is your anatomy, that is internal structure of a seed. Plumule develops into shoot, radicle develops into root, cotyledon, that is endosperm, that means nutritive value. It contains nutritive value. Then hypocotyl, hilum, seed coat or the testa, which is nothing but the covering. Here, in some seeds, the food reserves in the endosperm. I told you just now nutritive value जो होता है वो endosperm होता है. हाँ? So endosperm, the food reserves in the endosperm are partially used in the development of embryo. Of course, ये डेवलपमेंट के लिए एम्ब्रियो डेवलपमेंट के लिए न्यूट्रिशन प्रोवाइड करता है. Thus, the resultant seed is endospermic, and that's the reason we called it as this is endospermic, or it is also called as albuminous. For example, castor, coconut, maize. But in some of the cases, in other seeds, embryo absorbs food reserved from the endosperm completely during its development stages only. So in this case, पूरा उसको खा जाता है. पूरा वो क्या होता है? The whole thing, like the embryo only absorbs the food reserved from the endosperm completely during its developmental stages only. So here endosperm disappears in the mature seed, so the resultant seed is non-endospermic. Got it? And it is also called as X-albuminous. For example, P and Win. तो यहाँ पे एंडोस्पर्म रहता है. हाँ, यहाँ पे बिल्कुल भी डिसअपियर होता है. व्हाई? बिकॉज देयर इज कंप्लीट यूज ऑफ एंडोस्पर्म. हाँ, एम्ब्रियो जो है कंप्लीट ऑब्जॉर्व कर देता है वो एज अ फूड रिजर्व. So in this case, the seed is not endospermic. The cotyledons in some non-endospermic seed act as a food storage, and in other they are first photosynthetic organs. Micropyle persists as a small pore. I told you it is just the opening through which pollen grain enters. So in seed coat to allow the entry of water and oxygen during the soak. So development of seed and fruit development is triggered by hormones produced by developing seed. Yes, some internal factors are also responsible for this, for fruit development, and these are nothing but plant growth regulators. And plant growth regulators are also called as hormones. Here you can see the ovary begins to differentiate into fruit. differentiate हो जाती है, and ovary wall develops into pericarp, which is the outermost covering. Pericarp basically peel which get differentiated into fleshy fruit like mango, coconut. Understood? So significance of seed and fruit. Fruit provide nourishment to the developing seed. Fruit protects the seeds in immature condition. Seed serve as important propagating. What do you mean by propagating? Huh? Propagate to propagate. Production जैसे बोलते हैं ना हम, reproduction. Organs of the plant, seeds and fruits develop special devices for their dispersal and the help in the of the species also. जैसे सीड बैंक्स होते हैं कि पहले अवेलेबल थे ये प्लांट, अभी तो नहीं है, इट इज नॉट अवेलेबल इन मॉडर्न एज, लेकिन हमारे हम जैसे बोला जाता है कि हमारे टाइम में ये होता था, अभी नहीं है. So you will come to know different species were there with the help of seed bank if you preserve the seed. Understood? So that is also one of the significance of seed. You can preserve it to show this to your future generation. कि ये हमारे टाइम था, अभी नहीं है. Understood? Dormancy. Yes. Now what is dormancy? Inactive stage. It is inactive stage. It is a state of metabolic arrest that facilitates the survival of organism during the adverse environmental condition. Huh? Okay, Sejal, please tell me, have you heard about uh, hibernation, estivation जो animals करते हैं? कभी सुना है उनके बारे में? Animals hibernate, estivation and hibernation. Have you heard?
Yes, ma'am.
So what is estivation and hibernation? Hibernation is called as winter sleep, and estivation is summer sleep. Understood? To avoid the adverse, unfavorable conditions. Yes or no? Tell me. So in the same way, dormancy is nothing but it is a stage. Like in this stage, the uh, particular plant or we can say a particular species remains inactive. Huh? That means it is a state of metabolic arrest that facilitates the survival of organism during the adverse environmental conditions also. To avoid it, वो क्या करते हैं, डॉर्मेंट कंडीशन में चले जाते हैं, इनएक्टिव बन जाते हैं पूरे. हाँ, क्यों? क्योंकि उस इनएक्टिव कंडीशन में भी वो सर्वाइव कर सके. अंडरस्टूड? So some structural and physiological adaptive mechanisms for survival is called as dormancy. So some structural, we can say physiological adaptive mechanism for survival, we called it as dormancy. Matured and viable seeds will not germinate even in the presence of favorable conditions, and they are dispersed at different places during dormancy. Viable seeds germinate only after completion of dormancy period. हाँ, ये पूरा dormancy period खत्म होने के बाद ही seeds germinate होते हैं. Some examples of oldest mature seeds that have grown into viable plants किया है उन्होंने, ठीक है. Examples we have given. You can go through the examples. See, 2000 years back and 10,000 years. Lupinus articus and Phoenix dactylifera. Yes. Now only last part of this chapter is left. Apomixis. Then you have parthenocarp. Okay. Only few important terms are there. We will discuss it. Sejal, you need break? Tell me. If you need 5 minutes break, let me know. Because we have time, half an hour more.
No, ma'am.
Because I'm going to take class completely 2 hours class will be there, Sejal. So ऐसे मत बोलो कि मैम yes we have done with the chapter. Yes, of course I'm going to start with new chapter in this session only because half an hour is there now. And to explain apomixis and parthenocarpy, it will take 15 more minutes. So in last 15 minutes, I'm going to explain you about next chapter. Introduction will be there, right? So if you
Oh,
need 5 minutes break, after this I will give you. Let me finish this whole chapter first. Okay?
okay, ma'am.
Yes. Now, apomixis. A few flowering plants have evolved a mechanism to produce seeds without fertilization. We called it as apomixis. It is unusual sexual reproduction where there is no meiosis and syngamy. Syngamy means what? It is fusion of मेल एंड फीमेल गैमेट, लेकिन फर्टिलाइजेशन ही नहीं है तो सिंगैमी प्रोसेस भी नहीं होती है. Apomixis is the form of asexual reproduction. Can we consider, Sejal, if there is no fusion of male and female gamete, can we consider it as an asexual mode of reproduction? Tell me.
Yes, ma'am.
Yes, correct. Because see, mechanism to produce seeds produce होते हैं, but without fertilization. If it is without fertilization, we can consider it as a asexual mode of reproduction that mimics sexual reproduction. Now, the plant which shows apomixis term as apomictic plant. In apomixis, when a gametophyte organ or cell produces embryo-like structure without fertilization, is called as apogeny. And when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis, is called as apospory. So, gamy and spory. In this way, you can remember. For example, orange, mango. So, there are several methods of apomictic development of seeds development. For example, the one we have recurrent apomixis. What happens in this? The embryo sac is generally raised from either archaeosporial cell or from some other part of the nucleus. In diplospory condition, embryo sac is formed from megaspore mother cell. For example, Taraxacum. In apospory, the nuclear cells give rise to apomictic embryo sac. So it depends from which part what is going to originate. Understood? So based on this, like some other part of nucleus, so we have different conditions. Like in this condition, embryo sac is formed from megaspore mother cell. So based on which condition, uh, like embryo is going to originate, you have to understand this way, right? Which part is involved? This you have to remember. We have different parts basically. Then non-recurrent apomixis. In this type, megaspore mother cell undergo usual meiotic division and a haploid embryo sac is formed. Hence, here the embryo arrives either from the egg. Now, embryo जो है कहाँ से होता है? Either from the egg by parthenogenesis. Now you will ask me, ma'am, what is parthenogenesis? Here, this is again one of the conditions, Sejal, in which fruit is developed without the process of fertilization. पहले तो क्या होता है? Fertilization होता है. Then we have future organs, that means ovary develops into fruit, ovules into seeds. But parthenogenesis is again one of the condition where or in which fruit is developed without the process of fertilization or from some other haploid cells of gametophyte. The plants produced by this method are generally sterile. Of course, fertilization ही नहीं है तो क्या हो सकता है? And do not reproduce sexually. Adventive embryony. In this type, embryo may be developed from somatic nucleus, integument along with normal zygotic embryo. It is common in mango, orange, lemon, and it gives rise to a condition called polyembryony. See, the name itself gives you the meaning, polyembryony. Like this you can say, if you split the word, it is the development of more than one embryos. What is it? I'm so sorry. What it is? Yes. Polyembryony, that means more than 1, 2 or 3 embryos. When there is development of more than one embryos inside the seed, then the condition is called as polyembryony. Understood? Huh? So two main terms we have studied in this slide, parthenogenesis and polyembryony. Please write it down somewhere because these terms are very important. Parthenogenesis, the formation of food, or we can say the fruit is developed without the process of fertilization. And polyembryony, this is again one of the condition in which development of more than one embryos. We called it as polyembryony. So here separately we are going to talk about parthenocarpy, the process of formation of embryo without the fusion of gametes. That means in simple word what we can say, without fertilization. This is known as parthenocarpy. Fruits are formed without fertilization are called as parthenocarpic fruits. Such fruits either seedless or contain. Of course, because you know, if it is ovary, it develops into fruit. Ovules, ovary में तो ovules होते हैं, लेकिन it happens without fertilization. That means ovary and ovules are not there. So how it is going to develop into seed? So that's the reason parthenocarpic fruits are usually seedless or contain non-viable seeds. होते हैं लेकिन they are non-viable seeds. Okay, it occurs naturally in some varieties of pineapple, banana, papaya. You must have heard about it. In these plants, the placental tissues in the unfertilized ovary produces auxin. Now, have you heard about auxin? Auxin is plant promoting growth hormone. It is promoting growth hormone, auxin. Derived from indole 3 acetic acid, that is IAA, which is responsible for enlargement of ovary into fruit. So this auxin is, this is the hormone. We have different regulating hormone which will regulate the growth of particular plant. So auxin is plant promoting growth hormone. Then parthenocarpic fruits have higher proportion of edible parts than the normal fruits. So remember this term parthenocarpy. Yes. Now then we have polyembryony. Now what is polyembryony? Just now I told you, Sejal. What is it? Polyembryony.
Process in which more than 2 or 3 embryos are formed.
Correct. Very good. If you split the word into two words, now you will get the answer. It is the development of more than one embryo inside the seed. It is occurrence of more than one embryo in the seed, which further develops into a multiple seedlings. Of course, the additional embryos result from differentiation and development of various maternal and zygotic tissues associated with the ovule of seed. Polyembryony may be true or false depending upon whether many embryos arise in the same embryo set or different embryo sets are there. Have you heard about placentation? हाँ, 11th में आपने किया होगा placentation. हाँ, कितने ओव्यूल्स होते हैं, कितने एम्ब्रियो सैक होते हैं, कितने लोक्यूल होते हैं, how it is arranged. You must have heard about it. In adventive embryo polyembryony, an embryo develops directly from diploid cell of nucellus and integument. For example, in citrus. Cleavage is nothing but segmentation. जब segment होता है polyembryony का, zygote pro-embryo sometimes divide. See, cleavage is nothing but division only. Huh? So this pro-embryo divides into many parts or the units, and each unit then develops into embryo. Very simple. Cleavage is nothing but division or segmentation. So from this only you will come to know what you have to write in this. Polyembryony increases chances of survival of the new plant. Yes. So this is one of the advantages of polyembryony. Nuclear adventive uh, polyembryony is of great significance in horticulture. Okay. So understood. See, parthenocarpy can be induced artificially also, Sejal. Artificially भी parthenocarpy हम कर सकते हैं by spraying gibberellins, delaying pollination, then use of foreign pollens. In this way, artificially also we can induce parthenocarpy. So it is possible. This can be done artificially also. So I think we have done with the chapter. Now see, what 35 minutes, 35 nahi, 25 minutes are there. You can take 5 minutes of break, and then next chapter I'm going to introduce, that is reproduction in lower and higher plants. Hmm? Okay, answers and everything is given here. Yes. Understood, Tejal? Hmm?
Yes, ma'am.
Take 5 minutes break now. Hello, Sejal, are you there?
Yes, ma'am.
Okay, shall we start?
Yes.
Okay. See, let me introduce it first. Because in first chapter only I have discussed everything about reproduction. What is it? It is a biological process. Reproduction is necessary, but it is not essential. It is a production of a new organism of its own kind. Reproduction in animals also occurs mainly by two methods, asexual and sexual. Okay, now I'm going to share the screen. Hmm, Yes, is it visible? Visual, is it visible?
Yes, ma'am.
Okay. Mm, So, you know that there are two modes of reproduction, asexual and sexual. See, I'm not going to explain you again what are the importance of asexual and sexual mode of reproduction, because you know, involvement of one single parent, when there is fusion of male and female gamete. Already we have studied this. Variations किसमें मिलते हैं, फिर वो evolution में कैसे काम आते हैं, everything we have discussed. So asexual offsprings are produced by a single parent, no sperm, no egg needed. That means only single parent is involved. Here genetic stability is maintained. Why? Because there is formation of new individual of its own kind. Now sexual may involve the fusion of male and female gametes. From a male, one from male and one other from a female. So, which type of reproduction produces clone? Asexual reproduction or sexual mode of reproduction? It is asexual reproduction. Why? Because offspring are genetically identical. I told you before also importance of sexual mode of reproduction here, as only single parent is involved, genetic stability is maintained, right? Next generation में जो भी offspring होता है, it is genetically similar to the previous one, or the pre-existing one, or we can say of previous generation, that means parents, right? So, yes, fertilization, we have studied about it. It is the fusion of male and female gamete nuclei to form a zygote, single gamete, male gamete, and female gamete. Fertilization, zygote will form. So, zygote is the first cell that forms after fertilization. Hermaphrodites have both a male and female sex organs present in a single body. Earthworm, for example, here you can take hermaphrodite, have both male and female organs present in a single body. Now there are two types of fertilization: internal fertilization and external fertilization. Internal in reptiles, birds, and in mammals. External in most of the fish and the amphibians. Okay. Now, why is internal fertilization is essential amongst the land animals? Because no water in the animal's immediate environment to carry the sperm cells to the eggs. That is why internal fertilization is essential. Now, yes, not all eggs. This is not there in your syllabus. Yes. Here you can do comparative study. One parent needed, two parents needed, a male and female. No gametes required, gametes are required. Offsprings are genetically identical and offsprings are not genetically identical. So here you will get variation, Sejal. Why? Because as there is fusion of male and female gametes, here there is exchange of genetic material which results into variation. And you know, variations are needed or it is useful for the process of evolution, right? Yes, only one individual is needed to colonize a new area. A female and male are needed. A fast process, a slow process. Always increases the population, does not always increase the population. Very common among plants and simple animals. That means this is very commonly seen in lower organisms. And here almost all plants and animals, that means higher organisms we can consider. Advantage, a good characteristic shows up in all the offspring, and new varieties. I told you variations आते हैं इसमें, are produced which may be more able to adapt to the new environment. What is the disadvantages? A defect in the parent shows up in all offsprings. No variations occur. Here, excellent individual cannot give identical offspring. So it is exactly opposite. Now, gonads and gametes. I'm going to introduce before that one more. Uh, um, I think yes, asexual reproduction in animals. This also we have to study. It is very simple. In 10th standard, you must have studied about it. Asexual mode of reproduction. For example, budding in hydra, fragmentation in spirogyra, then regeneration. Like this. Have you heard about this? Hmm? Yes or no, tell me. It is not given in this slide, in this PowerPoint presentation, but I'm going to explain you. Give me only 1 minute so that I can show you. Wait, Rachel. Wait. Okay. Uh, yes. Is it visible now, Sejal? Something? Yes. Okay. Now, let's talk about asexual reproduction in animals first. Okay. Yes. Okay, asexual mode of reproduction. We just now we have done with comparative study. The offsprings developed by asexual reproduction are exact copy of her parents or their parents. They are genetically identical individuals, and hence they are termed as clones. Asexual reproduction is common in unicellular organism. That means it is commonly seen in lower organisms. In plants and animals having simple organization. For example, now here some of the asexual modes of reproduction they have given, we will just discuss and then we will stop. The first one is binary fission. Now binary fission, why the name is given binary fission? Bi means what? Two. Single individual divides into two equal daughter cells. The single cell organism of kingdom Monera and Protista reproduced by simple cell division. Simply by cell division it reproduces. We called it as binary fission. The cell divides into two halves, each half develops into new individual. Yes, in this you have in this diagram you can see. I think this diagram you have studied in 10th standard. Okay. This is irregular shaped amoeba. Then here you can see the single body, single parent divides into two equal daughter cells, and then it develops into new individual. Can you see here binary fission? That's the reason name is given. Bi means what? Two individuals. Now second, we have budding. I don't think so if it is given or not. Yes, it's given. Budding, bud. In East, the cell division is unequal and small buds. Bud means what? It is just a small outgrowth. Bud is nothing but small outgrowth. So, initially these are attached to the parent cells. Here you can see, can you see this small outgrowth here? And then this small outgrowth then after develops into new individual. In Hydra, there is small outgrowth, forms bud, and we called it as budding. It is a simple method of asexual reproduction, normally occurring in favorable conditions, and it is seen in a variety of animals like coelenterids, for example hydra, corals. Understood? So in hydra a small outgrowth is produced towards the basal end of the body, and then it develops as a bird which grows and forms tentacles. Tentacles are sensory in nature. Can you see? There are 5 to 6 in number. 1, 2, 3, 4, 5, 6. Tentacles. And develops into a new individual. This process is called as budding. The young hydra gets detached from the parent body. Then after, what happens? The whole outgrowth, it gets detached. The young hydra gets detached from the parent body, and then it becomes an independent new organism. So this is called as budding process. Okay. Have you heard about regeneration, Sejal? Regeneration. Have you heard about regeneration? What is it? For example, planaria. Huh? Have you heard about regeneration? What is it?
Yes, ma'am.
Huh? Yes. What happens in regeneration? आप बता सकते हो?
A body or a particular organ grows again.
Okay, that means removal of old part by new part. Can we say in this way? Huh? See, planaria, its cell becomes active and regenerates lost part or organ back to its original state. They can also reproduce asexually by fragmentation. Spirogyra जो example है fragmentation का. Also, it is seen in planariums that the anterior end exerts a pull on the posterior end, resulting a constriction on the middle part and splitting into two pieces. Here, no diagram they have given, but each piece then grows into new individual. Understood? Each piece grows into a new individual or new planaria. That means two clones of the original have been formed. Now, I think this is new for you. Have you heard about this before? Try to observe the slide. What is written? Tell me. What is it? We called it as asexual bodies. So what is it? Have you heard?
No, ma'am.
Okay. Textbook is there. Tell me. Please go through the diagram. Same diagram is given. Micropyle, which is just the opening, then inner membrane you have, then outer membrane, outer membrane, inner membrane. And can you see spicules here? Needle-like structures. Can you see?
Yes, ma'am.
Overall. And in this diagram also you can observe. Okay. So we are going to talk about gemmules now. These are the internal birds of freshwater sponges. Again, we are talking about lower organisms only. Understood? These are the internal birds of freshwater sponges. They resist drought, high temperature, freezing, and lack of oxygen. So, it is an internal bud formation or formed only in sponges. It has asexually produced mass or aggregation of dormant cells. We called it as, can you see inside dormant cells? We called it as archaeocytes. Archaeocytes. And these are capable of developing into a new organism. Can you observe this, Sejal? Inside it, we called it as developing cells or dormant cells. The archaeocytes get coated by a thick resistance layer or a secretion of amoebocytes. The gemmule is formed to overcome unfavorable conditions. Now, why there is formation of gemmule? To Yes, what is the reason? To overcome the unfavorable conditions. And on return of favorable condition of water and temperature, the gemmules hatch and develop into a new individual. For example, spongilia. Is it given in your textbook? See, in this diagram also you can observe spicules दिख रही है यहाँ पे, which is a needle-like structure. Understood? So, the gemmule is formed to overcome unfavorable conditions, but on return of favorable condition, जब उसको favorable condition मिल जाते हैं, that means water, temperature, then the gemmules hatch and develop into a new individual. For example, spongilia. Is it given in your diagram textbook, Sejal? Spongilia.
Yes, ma'am.
It is very simple. Now next we have vegetative propagation. I don't think so it is given in your syllabus, vegetative propagation. Till budding only they have given. And now human reproduction, male reproductive system and female reproductive system. This is not there, I think. Yes. Let me introduce what is sexual reproduction and then we will stop. Okay. So it involves the formation of male and female gametes. You know that in same individual, that is bisexual, or in different individual, unisexual of opposite sex. All organism reaches the stage of growth and maturity before they reproduce sexually. Now we have juvenile phase. In animals, the period of growth to attain maturity, we called it as juvenile phase. In plants, it is called as vegetative phase. In angiosperm, end of vegetative phase completes as soon as they start flowering. Yes. Then you have senescent phase. Now here it is again a reproductive phase. which is called as senescent phase, or it is a old age. Ability to become old, we called it as. Senescence means what? Ability to become old. After this, individual dies up. So we have different developmental stages. This is what the main point. Or understood? We have different developmental stages. Then flowering in plants, annual होता है, biannual plants. Flowering is once or twice in a year. In these plants, vegetative reproduction. What do you mean by vegetative reproduction, Sejal? What is it? Vegetative reproduction means what? Hmm? Sejal, yes, tell me what is vegetative reproduction?
Like, ma'am, it is done by cutting, grafting, or tissue culture.
No, that is artificial vegetative propagation. I'm talking about a natural, or when naturally it is takes place. You know that flower is considered as reproductive organ, but apart from flower, if any other body part, if any other plant organs involved in the process of reproduction, na, then it is called as vegetative reproduction. For example, if stem is involved in the process of reproduction, root, huh? Understood? Leaf sometime. So we called it as vegetative reproduction. In some plants, flowering occurs several times in particular season. In some plants, flowering occurs only once in a lifetime. After that, plant dies up. Hmm? Okay. So again, it depends. So sexually reproducing animals show various breeding patterns. Some, like the goat, sheep, and donkeys, are seasonal breeders, while humans and apes are continuous breeders. Okay, they can breed throughout the year, we can say. Okay. So I think clear, Sejal. Any doubt? Tomorrow, not tomorrow, in my next session, we are going to talk about, uh, एक second, let me show you. Not given everything. Okay, yes. We have discussed everything. Yes. So next lecture, we are going to talk about male reproductive system and female reproductive system. Okay, I hope chapter 1 is clear from your side. Still, if you have any doubts, you are free to ask me, because we have started second chapter, reproduction in lower and higher organism. Okay, Sejal? Hmm?
Yes, ma'am.
Yes, we will stop here now. Thank you so much. Bye bye. Good day. Stay
Thank
safe.
you, ma'am.
Bye.
Bye, ma'am.